Good morning, doctors. Today we will start with the influenza lecture. I'm Dr. Moheb, Associate Professor of Medical Microbiology and Immunology. Influenza is an acute respiratory tract infection caused by influenza viruses, and it usually occurs in epidemics. As you all know, we get influenza every year and also common cold. What is the difference between flu symptoms and cold symptoms? The common cold symptoms: influenza has higher fever, more intense headache and general aches and pains, fatigue and exhaustion. Both have stuffy nose, sneezing, sore throat, and cough is also more severe with the flu symptoms. What about the causative agent for influenza? Influenza viruses belong to a family of viruses called orthomyxoviruses. Orthomyxoviruses receive their name from their special affinity to mucous membrane containing surfaces, and they have no viremic spread. There are 4 antigenic types of influenza viruses, which are designated A, B, C, and D, based on differences in their ribonucleic nucleoprotein antigens. As we all know, antigenic changes are characteristic for influenza viruses. Antigenic changes continually occur within most commonly type A and to lesser degree type B, but type C is antigenically stable. Type D does not infect humans so far. What about the morphology of influenza viruses? Influenza viruses are spherical in shape. They have helical nucleocapsid. We all know now that nucleocapsid is composed of 2 parts, the nucleic acid. In case of influenza, it is RNA virus. It is segmented, single-stranded RNA virus. So the nucleocapsid is formed of the segmented, single-stranded RNA nucleic acids, to which proteins, of course, are attached to bind the nucleic acid together and the virus together. RNA genome of influenza A and B occurs at 8 separate segments. So the genome is not a single unit. No, it is divided into 8 segments. And each segment encodes for a certain protein, for a certain viral protein. As we see here, let's have a quick overview for the genome of influenza. We will have that 3 segments code for the RNA polymerase, polymerase basic 1 and polymerase basic 2 and polymerase acidic. Polymerase acidic is important because one of the drugs that you will take will act on polymerase acidic, so the drug will interfere with messenger RNA synthesis and thus viral replication. So 3 segments are responsible for the formation of the RNA polymerase. 1 segment is responsible for the nucleoprotein. The nucleoprotein binds the 3 segments together, and 2 segments are encode glycoprotein, which are surface proteins, hemagglutinin A and hemagglutinin and neuraminidase, the 2 projections on the surface that we will talk about in just 1 minute. And the 7th segment codes for the matrix. The matrix binds the nucleoprotein, the internal structures, the core of the virus, to the envelope of the virus. And lastly, a non-structural protein. So the 8 segments, each segment codes for a certain viral protein. The segmented nature of the genome is important cause of high reassortment that is exhibited between viruses. What is reassortment? It is the exchange. When 2 influenza viruses infect the cell at the same time, there can be an exchange of segments. So the virus A and virus B, when they infect the cell, a completely different virus C can exit the cell, which have combination of segments from virus A and virus B. So the nucleocapsid is surrounded by a lipid-containing envelope. So influenza is an enveloped virus. The envelope, of course, any envelope from for any virus, it is derived from the host cell membrane. But of course, it is modified, the host cell membrane, it is modified by the attachment of the viral glycoproteins, the surface proteins of the virus. The two virus-encoded glycoproteins, the hemagglutinin and the neuraminidase, are inserted in the envelope, and they are exposed as the spikes on the surface of the particle. Of course, the exposed parts are very, very important to the virus. They are responsible, as we will see, to the binding of the virus to the cell and reaching its destination. And of course, they are very important to the immune system because these parts are the antigenic parts to which the immune system forms its protecting or neutralizing antibodies. So changes that happens in this hemagglutinin and neuraminidase will determine the variation which will happen to the virus, and so it will affect the host immune response. So influenza A viruses are divided into subtypes based on these two surface glycoproteins. There are 18 subtypes for the hemagglutinin, hemagglutinin 1 to 18, and 11 subtypes to the neuraminidase, N1 to N11, and they occur, influenza A occur in all different combinations for hemagglutinin and neuraminidase. For example, H1N1, H2N2, H3N2, H5N1, and so on. Any different combination can occur and infect humans. So what is the function of the hemagglutinin spike? It binds the viral particles to the susceptible cell. It is the viral attachment protein which binds the virus to the cell receptors. It is the major antigen against which neutralizing or protecting antibodies are directed. The variability in this hemagglutinin is primarily responsible for the continual evolution, the changes that happen, and that can cause epidemics. It derives its name from its ability to agglutinate erythrocytes. As you all remember, there is an important feature for some viruses that have spikes that they can hemagglutinate RBCs. They can agglutinate the cells. Of course, influenza, one of the viruses in which it spikes, the hemagglutinin can agglutinate the RBCs. What about the neuraminidase? It is mushroom shaped. It is a tetra monomer. It is a tetramer, it is a tetramer. It is, of course, distinct from the hemagglutinin. The function of the neuraminidase, it functions at the end of the replication cycle to facilitate the release of the viral particle from surfaces during budding process. So it prevents the self-aggregation of the variant inside the cell. It helps with the release. It is also possible that it helps the entry of the virus through the negotiation of the virus through the mucous membrane in the respiratory tract to reach the target epithelial cells. So what about the changes that continuously happen in the hemagglutinin and the neuraminidase that are responsible for the antigenic changes that happen continuously with type A influenza viruses? There are two types of changes, either mutations, mutability, or as we have said, genetic reassortment, exchange when more than one virus infects the cell at the same time. They result in antigenic changes in the viral surface glycoproteins, and this is characteristic for influenza viruses. The two surface antigens undergo this antigenic variation independent of each other. So variation can happen in hemagglutinin or neuraminidase independent of the other glycoprotein. The two types of antigenic changes which occur are either antigenic drift, which are minor changes, or antigenic shift, which are major antigenic changes. So what is the drift? What is the antigenic drift? This results from accumulation of point mutations in the gene responsible for the hemagglutinin or the neuraminidase. So the mutation in the gene will result in amino acid change in the protein. This alteration in the antigenic site on the glycoprotein, a new strain will show minor differences from the strain of the previous year. This is why we get influenza every year, because there is minor changes that happen due to mutation. And why do mutation occur so frequently? Because the RNA polymerase lacks an important property, which is the fidelity, the proofreading. If any change happen, it will not be corrected. So mutations occur very frequently. So this new strain will escape recognition by the host immune system. Infectivity will persist because type-specific immunity is not entirely protective against these drifting strains, these strains which have minor changes, and this results in smaller epidemics every 2 to 3 years. For example, we have outbreaks of influenza every now and then due to these minor changes. What about the major antigenic changes? These occur at longer period, 10 to 40 years, and they cause worldwide pandemics, as we will see. They reflect drastic changes in the sequence of this surface protein due to genetic reassortment between human, swine, and avian influenza viruses. When human, avian, and swine infect the cell at the same time, any two of these three, or these three, or maybe more sometimes, there will be evolution of an entirely new subtype. This only occurs with influenza A, which undergo antigenic shift. Type B and type C are restricted to humans, so there is no incidence that other viruses will be in the cell in the same time. So how do we give a name to an influenza strain? Strains change from year to year, new strains appear every now and then. So how these strains get their names? First, we have to know the antigenic type. Is it type A, B, C, or D? Second, we give the host of origin. If the strain origin was swine, equine, swine of course from pigs, equine from horses, avian from birds, or from humans. If it is from humans, we do not put human in the name. So this strain, for example, does not have a host, so the host is human. Then we put the geographic origin. Then we put the strain number. This is lab number. The strain has been isolated in the lab and was given this number. And then we give the year of origin. And we give the hemagglutinin and the neuraminidase subtypes. And of course, this occurs only with type A. Type B and C does not have this hemagglutinin and neuraminidase subtypes. So for example, we have type A influenza, and the geographic origin was Hong Kong. This is the number of the strain and the year, and it was H3N2. This is influenza A from swine origin, New Jersey. This is the number of the strain, the year, and it was H1N1, and so on. Type A, it was isolated, or it has poultry, avian origin. It appears in Hong Kong. This is the strain number and this is the year. This is H5N1. This is the strain which caused the avian flu in 1997. If it is B, we will just put the origin, the geographic origin, the strain number, and the year. So what about the major antigenic shifts that resulted in pandemics? We will find that there is about 30 to 40 years, sometimes 10 years differences. So pandemics can happen from 10 to 40 years intervals. Of course, the major one was 1918, the Spanish flu, it was H1N1. In 1957, it was H2N2. In 1968, it was H3N3 in Hong Kong. In 1977, it was H1N1, the Russian flu. And in 2009, of course, you can remember this one, it was the H1N1 pandemic, which was formerly known as the swine flu. Okay? So what happened in 2009? The H1N1, the swine flu, a novel virus of swine origin appeared. It was a quadrupole reassorted. The genes came from four sources, four origins: North American pigs, Euro-Asian pigs, as well as avian and human influenza viruses. So it has four origins to get its 8 segments from. Okay, it was formerly called swine flu, but not anymore. Okay. What about the avian influenza, which happened in 1997, the H5N1? It was the first time that we document that humans are infected by an avian influenza virus. This is purely avian. The source was domestic poultry. And isolates have all their genes from avian viruses, indicated that this virus jumped directly from birds to humans. It crossed species. It was transferred directly from humans to infections through close contact. And these strains were not transmitted from human to human. Okay, so this strain H5N1, it did not cause pandemic. Why? Because for a pandemic to occur, a pandemic strain should be a new influenza strain. Okay, it was new. It caused serious illness. Okay, it caused serious illness, but it cannot, and it did not spread from person to person. So we cannot say that this is a pandemic strain. What about the currently circulating viruses nowadays? Currently, influenza vaccines contain influenza A H1N1 and influenza A H3N2. So why the vaccines contains these two strains with other two strains from influenza B? Because these are the currently circulating strains in the community. The H1N1 belong, of course, to the pandemic strain of 2009, H1N1. And it has gone small genetic changes over time. The H3N3 viruses, also the other currently circulating strain, go antigenic changes more rapidly, both antigenically and genetically. So the currently found strains are H1N1 and H3N2 and type B strains. Okay, and so the vaccine is tetravalent from these two type A and 2 strains of type B influenza. Okay, take care, flu influenza is ahead.